Hello everyone and welcome to another video of BioIVT's ADME 101 series. I am Tina Mueller, the Scientific Advisor for ADME Tox Services here at BioIVT, and today I will present ADME 101 CYP Induction. Let's start with a quick refresher of ADME as I have done for the previous ADME 101 series. So, as you probably remember, ADME stands for Absorption, Distribution, Metabolism, and Excretion. And understanding ADME characteristics is important in drug development as it helps to predict how a drug will behave once it is in the body. And today we will focus on metabolism and specifically what happens when the activity of drug metabolizing enzymes is affected. Before we dive deeper into CYP induction, let's look at the bigger picture of in vitro ADME studies and how CYP induction fits within the context of the common in vitro drug-drug interaction or DDI studies that are typically performed for small molecules. As we all know, the primary goal in pharmaceutical research is to develop drugs that provide therapeutic benefits while ensuring a patient's safety. In other words, we want to develop drug candidates that have desirable ADME characteristics. And in order to evaluate these ADME properties, drug candidates are routinely subjected to a series of in vitro DDI studies, of which the most common ones are listed in this table here. So generally, they can be grouped into three categories, namely drug metabolism, drug metabolizing enzymes, and last but not least, drug transporters. And today, we are talking about CYP induction, which is part of the drug metabolizing enzyme studies and assesses the potential of drug candidates to induce the expression of CYP enzymes and predicts in vivo DDI potential. Now, as the name of this video implies, we will focus on CYP enzyme induction today, but I wanted to mention that in some cases, UGT induction needs to be evaluated as well. So, what is the goal of a CYP induction study? In short, it assesses the potential of drug candidates to induce the expression of drug metabolizing enzymes and therefore predicts the in vivo DDI potential. This is important because, as you probably know, CYP enzymes are the most important class of drug metabolizing enzymes. And indeed, it is estimated that over 50% of all drugs are metabolized by this class of enzymes. So, if the activity of CYP enzyme changes, it can have a drastic effect on the metabolism of drugs that are metabolized by this class of enzymes. So the risk for drug-drug interaction is increased. In particular, if a CYP enzyme is induced and its activity has increased, this can lead to increased metabolism of concomitantly administered drugs, and as a result, their therapeutic effect might be reduced. Now, this simple figure illustrates that principle. On the left, we have the situation under normal conditions. A patient takes a drug which is metabolized, and in this example, let's say it is mostly metabolized by CYP3A4, and the exposure, clearance, and efficacy is as predicted and intended. Now, if the patient takes this first drug, so drug 1, as well as drug 2, the situation might change. Let's assume that drug 2 is a CYP3A4 inducer, which leads to an increase of CYP3A4 enzyme activity, and as a result, drug 1 will be metabolized faster. That means that there is a shorter exposure of the drug in the body, and therefore efficacy is likely reduced. So I hope I made it obvious that understanding a drug candidate's induction potential is critical and supports the evaluation of the risk for DDI. Now, regulatory agencies understand that obviously as well, and evaluating CYP induction for CYP1A2, 2B6, and 3A4 is required by the ICH M12. In addition, If CYP3A4 is shown to be an inducer, it is recommended that CYP2C8, 2C9, and 2C19 induction is also assessed. Now, I also wanted to show two classic examples where CYP induction has played an important role in drug-drug interactions. The first example is around oral contraceptives. So estrogens and progestins, the two active compounds in oral contraceptives, are primarily metabolized by CYP3A4. If a patient also has to take the antibiotic rifampicin to treat gram-positive or mycobacterial infections, so for instance, tuberculosis, she needs to be cautious. Rifampicin is a strong CYP3A4 inducer, resulting in the hormones of the oral contraceptive being metabolized faster, and this will lower the efficacy. Oral contraceptives are also drugs with a so-called quantal dose response. So if the levels drop under a certain threshold, they are going to be ineffective. This might lead to unwanted results, such as an unexpected pregnancy. Another example is St. John's wort, which is a natural supplement used for different indications, such as mild depression. The substance hyperforin in the plant is a strong CYP3A4 inducer, and as a result, St. John's wort is known to be causing DDI. One of these is with cyclosporin, which is an immunosuppressant and often given to organ transplant patients. Analogous to the previous example, if St. John's wort and cyclosporin are taken at the same time, it causes cyclosporin to be metabolized too quickly, and as a result, it weakens its therapeutic effects. Namely, it weakens the immunosuppression, and which can lead to a possible transplant rejection. Let's now take a minute to understand the general mechanism of CYP enzyme induction. In this context, we will focus on the most common mechanism, namely transcriptional gene activation via nuclear receptors. For CYP enzyme induction of CYP1A, 2B, and 3A, there are three important nuclear receptors. First, the aryl hydrocarbon receptor, or AHR, that will induce CYP1As. Second, CAR, the constitutive androsten receptor acting on CYP2B6. And last, PXR, or the pregnant X receptor, inducing CYP3A4, as well as the 2Cs, so 2C8, 2C9, and 2C19. Please also note that this is a simplification and that there is crosstalk between CAR and PXR with overlapping induction of CYP2B6 and 3A4. Now on the right, this simple scheme shows how CYP enzyme induction works in principle. Both the drug and the receptor are in the cytosol when the drug binds to the receptor. The complex then enters the nucleus, where it will ultimately bind to specific DNA sequences, thereby inducing gene transcription. That will result in more CYP mRNA, CYP protein, and eventually in an increase of CYP activity. Now, as a side note, these processes take a little while, so the time frame here is days. Let's now switch gears and look at this table, which shows the typical design of CYP induction studies, which closely reflects the setup here at BioIVT. There are two study designs depending on the objectives. The first is a screening, or probably more accurately called compound ranking study, and the second is the definitive or IND-enabling study that meets the requirement for regulatory submission. For both of these designs, CYP induction studies are performed in single donor human hepatocytes. And for compound ranking, one donor is usually sufficient, whereas for IND-enabling studies, 3 individual donors are typically evaluated. The treatment time with the drug is either 24 or 72 hours, and for the longer treatment time, the drug candidate is added every 24 hours. Typically, we recommend 7 concentrations and 3 replicates. And of course, proper controls are performed. 1 solvent control and negative and positive inducers are included at 1 concentration as shown here. These are the prototypical inducers. So we have omeprazole at 50 micromolar for CYP1A2, so the AHR receptor. For CAR and 2B6, phenobarbital is used at 1000 micromolar. And rifampicin is the typical inducer for PXR and CYP3A4, as well as the 2Cs. The negative inducer is flumanesil at 25 micromolar. Moving on, regulatory guidance recommends evaluating CYP induction by measuring mRNA levels using qRT-PCR for CYP1A2, 2B6, and 3A4. With that said, enzyme activity can be measured as well, but if the drug candidate is also an inhibitor, it might mask the results. Now, in the IND-enabling study design, a concurrent cytotoxicity assay is carried out. The cells are assessed for lactate dehydrogenase or LDH release at 3 different time points, which is a common method to evaluate whether the cells are still healthy or whether the drug starts to kill the cells, especially at the higher concentrations. Now, if that were the case, it would need to be taken into account when interpreting the results. Also, the ICH-M12 guidelines require the analysis of the spent media over several time points. Here at BioIVT, we do 4 time points on the last day of the experiment. This is to demonstrate that the cells were exposed to the drug at the concentrations intended, and that analysis is done by LC-MS. Last, the deliverable for CYP induction studies is a report. It will show the fold induction and, if possible, the EC50 and Emax, 2 parameters which we will talk about in a couple of slides when we look at the data. I also wanted to mention that for the IND-enabling studies, the report is ECTD compliant when you do the study here at BioIVT. Now, I have mentioned that CYP induction for CYP1A2, 2B6, and 3A4 is assessed using mRNA levels. Now, if CYP3A4 is shown to be an inducer, regulatory guidance requires that the CYP2Cs, so CYP2C8, 2C9, and CYP2C19 are also evaluated. With that said, the induction potential for the 2Cs is typically lower than that for CYP3A4, and we often recommend using enzyme activities to assess the 2Cs. Indeed, it is required to use enzyme activity for 2C19 according to the ICH-M12. Now, let's look at some typical data of CYP induction studies and how to interpret the results. We have here 2 bar graphs that show a typical positive and negative response. The fold change is on the x-axis, and different concentrations are shown on the y-axis. On the left, we have an example of an inducer with a positive response. So, with increase in concentration of the drug candidate, the fold change increases. In contrast, the plot on the right does not show induction and has a similar fold change with increasing drug concentrations. Also, the fold change is much lower than that of the positive control. As you can see also in both graphs, the controls are shown for comparison as well. Now, the ultimate goal is to interpret these data in order to assess whether there is a DDI risk due to CYP induction. The simplest way of how to interpret the data according to the ICH-M12 is a method called basic mRNA fold change. The ICHM-12 expresses the criteria as a negative and states that in vivo induction cannot be excluded if both of the following two criteria are fulfilled for at least one of the three donors. First, the increase in mRNA expression of the CYP enzyme analyzed is dependent on the concentration of the drug candidate. And second, the change in CYP mRNA expression is greater than or equal to twofold at drug concentrations that are lower than or equal to 50 times the maximal unbound concentration. I would like to emphasize again that in IND-enabling studies, 3 individual donors are evaluated and only one of them has to show or has to meet these two criteria. Here's another example of the type of results that are generated in the CYP induction study at BioIVT. This is an example with rifampicin for CYP3A4 induction with 3 individual donors, and the treatment time was 72 hours. And this time, the data was plotted with the x-axis showing the drug concentration, and on the y-axis, the effect as measured in fold induction. As you can see, the fold induction increases with increase in concentration, and rifampicin, as expected, induces CYP3A4. Now, importantly, 2 parameters can be calculated from these curves, and these are Emax and EC50. Now, E measures the effect of the tested drug, and with that, Emax is the maximal induction effect of that drug. This is expressed as fold change compared to the vehicle control, so the sample where no drug was added. EC50 is a concentration, as can be seen where the red dashed line intersects the x-axis, and it shows the concentration of the drug at which half maximal induction is seen. Now, let me very briefly mention what follow-up in vitro studies can be considered if in vivo CYP induction cannot be excluded using the simple method which I just showed. First, and without going into too much detail, there's the correlation method. It is based on the relative induction score of well-characterized hepatocyte lots and compares the induction effect of the drug candidate to that of established in vivo inducers of the CYP enzyme that was induced. With that, in vivo induction effect can be predicted. Second, the basic kinetic model can be used to calculate the so-called R value. Now, simply said, the R value can be calculated using this formula here that includes the Emax, the EC50, and the unbound maximum concentration in plasma. And last, D is the so-called scaling factor. Now, to make this short, if R is greater than 0.8, the risk for in vivo induction can be excluded. With that, let me summarize 4 takeaways for CYP induction studies. First, CYP induction studies evaluate the potential of a drug candidate to induce the expression of drug metabolizing enzymes and predict in vivo DDI potential. Second, the evaluation of the potential for induction of CYP1A2, 2B6, and 3A4 is recommended by regulatory guidelines such as the ICHM-12. If CYP3A4 is induced by a drug candidate, the 2Cs should also be assessed. Next, CYP induction potential is typically assessed in platelet hepatocytes in individual donors. The endpoint is mRNA analysis, with the exception of the CYP2Cs, for which we recommend enzyme activity. And last, the results and the conclusions of CYP induction studies will aid in understanding of the DDI potential. They can also assist in the design of clinical DDI studies. And with this, we are at the end of this short introduction into CYP induction studies. And here is a quick overview of the BioIVT website for the in vitro ADME products and programs. Please take a look to learn more, download resources, and of course, talk to us or request a quote. Thank you so much for watching, and please reach out if you have any questions, or of course, if you have any needs for a CYP induction or any other in vitro ADME study. Thank you. Bye.